are some really basic concepts of vision. We all know from, let's say, middle school or from the primary school that what is happening in the eyes is that we have a lens, and then there is a kind of let's say, an image from the actual kind of object outside is happening on the back of the eyes, and we perceive that. So that is something that really basic. And we have started to learn in high school that, okay, we have what we have in terms of a kind of series of neuron-like cells that are in retina, and they receive information. So what you can see here, at least we have simply 3 layers. We have some sensors here that are working. We have some bipolar cells here, and then we have some ganglions. The interesting part is the photo beams would go from this direction. So their location is that basically the sensors are behind. So the light beams would go through these layers, and they go to be able to touch 2 types of cells that we have. So we have rods and cones. I'm sure you have heard about those types of cells. We have something like 120 million rods and something like 6 to 7 millions of cones. So there are far more in terms of the number, something like 20 times more than the cones. Okay. There are some experiments to just show you some of the specific defects that we have in the visual system. This is one of those experiments. Before kind of showing you the experiment, let me just go back and show you a few other pieces that we have here. So we have one central point that is in the axis of your eye that we call fovea. It is the most precise part of the visual field, and we have huge amount of cones here, and cones are responsible for color vision. But in the periphery we have mainly rods that are responsible for basically night vision, or when we have very little amount of brightness in the environment around us. So when we have a low level of illumination, then we will have rods working for what we call night vision. So they work on the periphery. They are not very much precise, but the good thing is that they can work with a really little amount of illumination. But for the cones, they need a huge amount of illumination, but they are colorful and they are more precise to be able to see different things. But we have another item that is also important. This small point here is called something like a blind spot. Why? Because this is exactly the part that these neurons, these axons, are leaving the eye. So they just go into the optic nerve to make optic nerve. And so this specific part does not have any of these sensory kind of items that we can see here. So we cannot, there is no photoreceptor here to be able to kind of receive visual information. So this point is blinded. And as you can see here, there is something like a small degree of the visual angle differences between the fovea and this blind spot. Okay. If we start to do this experiment, you are supposed to see the cross. Okay, see the cross and try to go just in front of the cross as much as you can, go to the really close part of the cross, and then you are supposed to come back from the cross. And when you meet that specific visual angle in a way that the cross, because when you are focused to the cross would be on your fovea, so it would be on the center point on the fovea that I have mentioned before. And then what I'm expecting is to have that red point on your blind spot. So depending on the degree, most of the time the blind spot is a little bit kind of lower. So you need to have that kind of red spot a little bit up as well. So it should be something like in this direction. When you are focused to this, in one point, that red dot would go exactly over the blind spot, and you start not to see that. So if you do experiment the right way and try to different direction of your head, you would experience that. When you go to a specific kind of distance, you can see that for a specific distance and a specific angle, when you are focused to the cross, you cannot see the red dot. Okay, so that is exactly the time that red dot is represented over your blind spot, while your cross is right on your fovea. Okay, so that is an experiment you can experience when you do the test. And it's quite interesting that if you do it with this grid or something like a sets of lines or parallel lines, when you do this with these parallel lines, what you will see if you do this. I'm not sure if anybody would be able to do it right now with the computer screen, but you can try it. You can try it if you can see that or not. If there is anybody who is able to get to a specific kind of angle and kind of distance in a way not to see the rod, and you need to close one of your eyes. I mean, it's going to be done with one eye, and depending on the direction of the red dot, you need to kind of close right or left eye, because what we have, those kind of blind spots are located a little bit to the medial and down in terms of the center of your fovea, because the fovea is here, but those spots are a little bit down and medial to that. So you need to think about, okay, if I have that in the right side, I need to have that with my kind of with the eye, that this is going to be reflected onto the let's say blind spot. So it's going to be a little bit complex in terms of kind of the direction and the point, but it's totally doable. The interesting part is that when you put this red dot over your blind spot, the interesting thing that is happening is that you can see the entire kind of set of these lines without any gap. So what you will have is that it seems that the brain is just making the lines that you cannot see on your brain, on your blind spot. And that's a really interesting part of your vision. We see environment around us. We do not see any blind spot in the environment around us. But in reality is that those blind spots are being covered by your brain. So that is how the brain is processing that information. So it basically kind of fill the gap. And if the gap is about the lines, basically it covers the lines that you have in between, and you feel that there is nothing there, it's just those lines. So it is not something like the kind of just a white spot or the dark spot in between. No, it is just the real dots, so the real lines. So it basically make the lines by making the gap filled with what you are expecting to see. So that's an important kind of experience in terms of getting sure that your brain is able to make what is happening, what is expected to be seen, in a way that you would see the environment around you without having any gap inside. Even you know that there are some gaps in your visual field. The other thing that is going to be critically important is that what we see in the visual system is just we are making a representation from the environment to our eyes, and then from eyes to the brain. So we have what we call a representation from the environment to the eyes, to the retina, to the neurons that we have in retina, and from that to your thalamus nuclei that are related to vision, and then from that to your visual cortex. So we will have representations of this environment in different levels. And we have this representation in different parts of the visual cortex and visual processing areas. So in each area we have a new representation. And in each kind of phase we have representations with different features. So we'll discuss about how these representations are going to be different. Okay. So going back to what we have been discussing about, this is a more, let's say, in detail figure discussing about how these rods and cones are interacting. I'm not going to go to the details in terms of how these photoreceptors are working when we have photons hitting these kind of sensory parts, and what are the processing that are happening to have an action potential from here to go from here, and then from here to the ganglion level. So these are basically, this is the bipolar, this is the sensors. So photosensors, bipolar neurons, and then ganglions. And these pieces are not just to connect sensors to the ganglions. They are processing information. So the information, the level of information that we have here, that is the reflection from the environment, the level of information that we have in the sensory neurons, the level of information that we have in the bipolar neurons, and the level of information that we have in the ganglions, these levels are totally different. So I will discuss about how these levels are different from each other. And why do we name them as bipolar? And there are different types, of something like 4 different types of bipolars, and there are some transverse neurons, and they have specific roles. I'm not going to go to the details about how these circuits are working, but it's amazing, and there are people who are just specialized in processing these circuits at that level, and how these circuits are processing information in a way that at the end, for example, you have, let's say 1 or let's say 40 gigabyte of information, but the same image that is going into the optic nerve, it is something like 1 gigabyte of information. So it basically, these layers are compressing the information in a way that we can have that information in the optic nerve. Otherwise, you could not have the representation of all these neurons going through the optic nerve. And in the optic nerve, we have something like 1 million fibers that are crossing and composing the optic nerve. We cannot have the entire amount of, let's say, I told you we have 120 millions of rods here, 120 millions, and something like 7 millions of cones here. So all of them could not have signal to the brain. So we need to process the information in a way that when we have the information in the optic nerve, it should be compressed. It should be something like 1/40th of the information that we are receiving in the retinal level. Otherwise, the information could not be transferred to the brain. So there are many processing that are happening here. Okay, to give you a sense about what are the processing that could be happening there, let me show you some other networks that are working in that level. One of the processes that are happening in the brain is that the information that we receive, the processes that are happening in the eye, we are not talking about the brain, we are still talking about the eyes. The processing of information starts from the eyes. The information that we receive is in something like a bitmap format. So we have the information of every single pixel in this photo. So we know what is the contrast, or what is the amount of light that we have in every single bit or pixel of this information. So we call that bitmap format. Okay? But to compress the information, what we do, we say that, okay, I define this area, I just define a surrounding kind of box, and say this box is gray like this, gray, let's say 20% gray. So instead of just reporting every single dot of this part, I'm just reporting the edges, and then what is happening inside the edges. So that is the way that the information is being processed from single pixels to some shapes with edges. And this is something like what we do with JPEG format. So technically the retinal circuits would change the image in a way that it would be compressed. So we already have the level of information processing in the retina that are happening. So let me just give you more kind of details about how these kind of edge perception or processing that are happening in just a really kind of short time that we have. But to give you a sense how these neurons are processing really complex information in a most, let's say, efficient way. Because in a really small place, how these sensors could compress the information in such an efficient way that we'll discuss about. Okay, so what we have, let me show you another part of that. So what we have, most of the time we have what we call receptive fields. So for each ganglion, okay, for each ganglion we have what we call a receptive field. So this is a central ganglion, okay. We have one central ganglion, which is going to have one fiber to the optic nerve from one of those 1 million fibers in the optic nerve, okay? So this ganglion is going to contribute just one single fiber to the optic nerve. And then these are the sensory, most of the time rods and cones, most of the time these are the rods, and then the bipolar neurons, okay? So we have something like a kind of architecture, neural architecture like this. And these receptive fields are like this. They are called center positive surrounding negative receptive fields. So we call them receptive field, okay? In these receptive fields, if I make an illumination in the center of that area, so if I just make a light to the center, if I make a light to the center, we have a firing. If I make the light bigger to just cover the entire kind of center, we have highest level of neural firing. But if I go beyond that, the level of firing would go down. Why? Because we are stimulating these ones, and these ones would have negative impact on the central ganglion, okay? And if I go up, then the level of neural firing would be really low, okay? So this specific receptive field is just perceiving one bit of information here, but not information in other parts. So it is really selective to this kind of illumination in one single dot, not the illumination is going to be bigger or very much smaller. So it just really kind of specialized for that specific, detecting that specific dot in the visual field, okay? So we call them receptive fields, and how they are positive and negative, and how these really simple networks are processing the information in such an efficient way. And what I really like about vision system is one of the best ways to learn about how these neurons are coding information, how the information is coded in this system. It is more complex in other sensory systems, but the vision system is more well-known and it is well established, so we know that how these kind of networks are working. So you can start to have a sense about how these neurons are processing information. And then let's see these grids that we call Hermann grids. When you see these grids, most of the time, those who see these grids for kind of just few seconds, they feel that areas in the middle, they are a little bit darker compared to areas that are here. And the reason that we have this one, let me just make a kind of bigger grid for you. The reason that you have a feeling that these areas are a little bit darker is that when we have a receptive field here, when the receptive field is located here, you have the representation of these visual fields over your retina. This one would give some negative feedbacks to the center, but this one would give more negative feedbacks to the center. So this one, the firing rate would be something like, but this one would be firing like this. And that's the reason that we see these parts brighter than these parts. And it depends on your distance to the grid and some other factors as well. But you can see that this really simple visual illusion is something that is related to how these receptive fields are registered to these kind of representations from the environment to your retina, and how these things are being processed by the simple circuits that I told you, and how these parts are going to be perceived as darker parts, okay? So this is how the information is being processed. Let me just give you a few other examples to give you a better sense about how these things are working. So this is a receptive field with on-center field. So it is processing the information in the center. If I give an overall diffuse illumination, so this is the time that I have the illumination. So this is dark illumination. So with or without illumination, the firing rate is not going to be different, okay? So illumination, when the illumination is filling the entire receptive field, it's not going to make a change. But if I make the illumination in the middle of the receptive field, this is what I will see. So there will be an increase in the firing rate of this central ganglion. Okay, so there is an increase. If I do on the other side, if I do just annual illumination, if I just illuminate outside without illuminating in the middle, what I'm going to see? What are you expecting to see? There is a suppression. This is called lateral inhibition. So these parts are laterally inhibit the center. So there is no firing right now. If I had a small spot in the middle or having a small spot outside, this is the effect. So if the spot is in the middle, there is a little bit of increase. If there is a spot outside, there is a little bit of decrease in terms of the response. And then these visual fields, if I have, instead of on-center, if I have off-center, and by off-center I mean they are going to have positive effects, and this one is going to have negative effects. So we just change the type of synapses that we have here. If I have the same, as you can see here, with dot in the middle, there is a kind of suppression. Outside, there is a kind of increase in the firing rate. So you can see how these things are working. You need definitely time to have a kind of better perception about these neural coding, but it is a really good opportunity to have a sense about how these neurons and excitatory and inhibitory synapses are going to code information in a way that make kind of a larger amount of information to a smaller pieces of information in a specific format. Few other examples. Sometimes we have a higher level of information. So we have some receptive fields that are working together. They are related to some lateral geniculate nucleus in the thalamus, and then V1, which is the occipital cortex. So this one is the primary visual cortex, thalamus, and then retina, okay? And these parts are direction sensitive. So this specific neuron would have high activity when we have a line like this here in the middle. But if you have a line in this direction, the level of response would be, if you remember the cat kind of experiment that we discussed about before, so you can see that how these lines are related to this selectivity in the V1 level. So this is not about the selectivity in the ganglion level. It's about selectivity in the V1 neuron. So that's the reason that we have specific neurons in the visual cortex that are sensitive to the direction of these light beams. So that's going to be critical to have a better understanding about how these direction-sensitive neurons would be functioning in the occipital cortex. Okay, so that is the end of this part.